Hi, class, and welcome to Lab 11, which will be on mitosis or cell division. The lab worksheet that you're filling out has different links associated with each activity, and each link to the activity will take you through kind of that part of the worksheet. So your worksheet should be pretty simple and easy to follow. I'll give you a brief introduction into mitosis and cell division right now. This PowerPoint is also posted on Canvas. So within the life cycle of any cell, there's the development phase, or where a fertilized egg will become a new adult. And there's also the reproduction phase, which is the formation of new individuals from pre-existing ones. And that's what we're talking about now, this cell division. Sexual reproduction is the union of one egg and one sperm, and asexual reproduction is the continuation of a species solely from one parent. The original cell is always called the parent cell, and the resulting cells are always called daughter cells. So as we're talking about cellular reproduction and cellular division, the parent cell will be the original one, and the cells that result after cell splitting are the daughter cells. In cell division, chromosomes are always duplicated and the cell divides, and each daughter cell is genetically identical to the parent and the other daughter. So this is in cellular division. So here's a look at a parent cell. First, the nucleus divides as those chromosomes or genetic material divides. Then the cytoplasm divides, and eventually we get two new daughter cells. And this is a really cool photomicrograph of the division of cells, showing how a new plasma membrane and cell walls forms right down the middle after the nucleus divides. Cell division, sometimes called mitosis, can be used as a mode of asexual reproduction because you have one cell splitting into two. For the development from a single fertilized egg into adult, as more and more cells are created, or as the replacement and repair of cells, as cells need to be divided and replaced. So for example, all your skin cells on your skin are constantly being sloughed off, and they need to be constantly duplicated and divided to replace damaged skin cells. The chromosomes of eukaryotes, those are complex cells, will duplicate or replicate, so make another copy of them with each cell division. And this is a look at your DNA. So your chromosomes are made up of DNA, which is your genetic material. And DNA is made when the two kind of wound ends of the sugar phosphate backbone are connected together with what we call base pairs. And these base pairs are always complementary. So for example, thymine always pairs with adenine. Guanine always pairs with cytosine. So you can see here how these base pairs are connected with hydrogen bonds. This is a look at how the base pairs are connected, forming this DNA, which is this wound helical structure. The DNA double helix winds around proteins called histones to become tightly wound until eventually we get to this structure, this X-shaped structure called a chromosome. And a chromosome is made up of two halves, which we call a sister chromatid. So each half of this X of the chromosome is called a chromatid, and the chromosomes are what will be pulled apart or separated as the nucleus will separate right before the cell divides in mitosis. So here's a look at some more chromosome terminology. This is an unduplicated chromatid and then a duplicated chromatid, and we have sister chromatids together to make up one chromosome. The centromere is the center part that holds the two sister chromatids together. And then the genes, you can see the kind of the bands of different colors. A gene is just a section of DNA within a chromosome, and you can see they're colored differently there. So each gene can be a different section of DNA on the chromosome. Eukaryotic DNA, most of the time, the molecule is in the form of thin, loosely packed chromatin fibers. They will condense into visible chromosomes just before the cell divides. So normally, your DNA is in the nucleus of your cells in just these small, tiny chromatin fibers, but right before any cell divides, the chromatin will condense more kind of compactly into these visible chromosomes. And these visible chromosomes are formed just before cellular division. And this is a look at this is the phase, what we call prophase, showing how the chromatin, the DNA strands, are becoming tightly more compacted together to eventually form chromosomes. And this is a look at interphase. And interphase is the period of a cell cycle, which is just normal cell growth. So inside the nucleus, you can see the chromatin. You don't see any chromosomes forming yet because the cell is not going to divide. Right before the cell does divide, the chromosomes will replicate or make another copy. The sister chromatids are joined together at the centromere, which will create a replicated chromosome. And when the sister chromatids separate, these are called chromosomes. The cell divides into two daughter cells, each with a complete and identical set of chromosomes. So we have one chromosome and one replicated chromosome made up of two sister chromatids attached together at the centromere, and then two daughter chromosomes at the end of cellular division. So here's a look at a chromosome underneath a photomicrograph. Pretty cool to see that in real time. The cell cycle, then, most of the time, a cell is in what we call interphase part of the cycle. G1 is when the cell grows in size. The S phase is when DNA synthesis or replication occurs, so when the DNA makes a copy of itself. G2 is when the cell continues to grow and prepare for cellular division. And eventually we get into the M phase or the mitotic phase. And this is where the cell contents will divide via nuclear division. The duplicated chromosomes will be evenly distributed into two daughter cells. And the word cytokinesis has to do with what we call cytoplasmic division of the cell. And here are the different phases of mitosis. Prophase is when the nuclear membrane breaks down, the chromosomes become visible and a spindle starts to form. Metaphase is when the sister chromatids line up along the middle of a cell. Anaphase is when the sister chromatids will separate. And telophase is when the cell starts to divide and a nuclear membrane forms. So here's a look at the cell cycle. Again, about 90% of the life of the cell is just spent in normal interphase. And that just means that normally cells are just carrying on their average everyday activities. And about 10% of the time, they go through the M or mitotic phase when they actually divide into two daughter cells. In animals, sorry, let me get rid of this so we can actually see this. In animals, cytokinesis differs for plant and animal cells. So in animal cells, a cleavage furrow develops, which is an indentation right when the two animal cells will divide. And in plants, the vesicles will fuse into a membranous cell plate. So, and that cell plate will eventually form a new wall between daughter cells. So this is a look at the cleavage furrow that divides in animal cells. It's kind of like an indentation right before the cell will separate. There will be kind of an indentation of the plasma membrane. Whereas in plant cells, a cell plate will begin to form. And that cell plate will eventually just form a new cell wall between the two daughter cells. Here is a look at different examples of cells in different stages. So here's an example of a cell in prophase. Whenever you see the chromosomes lined up straightly there, that's called metaphase. Anaphase is an example right here when the chromosomes are being pulled apart. And then telophase and cytokinesis, you can see here because you can see a new plasma membrane starting to form between the cells. So here's a look at a plant cell undergoing, you can see the chromosomes. In a slide example like this, you can see examples of all stages in one slide. So this is a stage of prophase, anaphase, the chromosomes are being pulled apart. You can see a new cell wall forming here at the end of the mitosis stage in plant cells. And here's a good example of the two daughter cells that have formed in an animal cell, for example. And here is a plant, for example, undergoing mitosis. In mitosis, the parent cell will start with a replication of DNA, and the DNA divides into two cells during cytokinesis, which means the splitting of the cell. and it will result in two daughter cells that are exactly like the original parent cell. A parent cell has 12 chromosomes, and the cell undergoes mitosis. How many chromosomes will be in each daughter cell? There will be 12 in each daughter cell because remember, the parent cell will completely duplicate its DNA before it divides. So there will be 12 chromosomes in each daughter cell, and the two daughter cells will be exactly alike. You can observe pictures of animal cell mitosis using this whitefish embryo pictures. Don't worry about this. You can just ignore that. And for these activities that are on your worksheet, just follow the instructions that are on the worksheet. And then there's a couple of resources at the end if you want to take a look at some more animations. And thanks for listening, guys.